Okay, I hope everybody can hear me fine. Welcome to this webinar on pupillometry. I'll start with just a quick outline of what we're going to cover in this webinar. Unlike the other webinars, which were more focused on practical aspects of things, looking at experiment builder and data viewer, this is more going to be a theoretical webinar covering the kinds of considerations you should bear in mind when doing pupillometry research, as well as some of the sort of practical considerations for things like experimental design, and also some practical things like how to actually extract the pupil data itself, look at it and convert it to millimeters. And I'll cover at the end various analysis approaches. I'm going to start off with some basic background information about why pupil data is interesting, covering some of the history of pupillometry research, and also very, very briefly some of the physiology and pharmacology, mainly because this has implications for how we interpret and understand pupil data. I'll then talk in a bit of detail about many of the things which are well known that influence pupil size that probably aren't the things that you're interested in when you're doing a study that's specifically measuring pupil size. For example, things like luminance levels and blinks. And as we'll see when we actually analyze some data towards the end of this webinar, these can have dramatic effects, and they need to be borne in mind when you're looking at your data. I'll also talk about the pupil foreshortening effect, which is another important thing to bear in mind. And all of those things are going to feed into a discussion of the kinds of experimental design considerations you might want to make if you are going to plan or do a study measuring pupil size. And then finally we'll get on to the practical stuff at the end, where I'll show you how to do the conversion and how to analyze the data itself. So it's been known for a long, long time that the eye itself is a useful window onto the mind. And one of the reasons people use eye trackers is so that we can capture some aspects of this, which is where people are looking, what they're focusing their attention on. But it's been known equally long that the pupils themselves can be an important clue as to underlying cognitive operations. So just as we can look at the shape of the eyes, if you like, in these images here, and I think I can tell that's probably fear, that's probably anger, that's sadness, so that's probably happiness. So we can derive information just based not just from where the eyes are looking, but from the actual eyes themselves. And this has been known for a very, very long time. In fact, there are quotes as far back as the 16th century showing that people have noted the relationship between pupil size and cognitive or emotional state. So William Harvey wrote, "In anger the eyes are fiery and the pupils are contracted," which is interesting because it's probably wrong. He was a physician and a scientist, whereas Charlotte Bronte wrote, "Pupil shame, ire, impatience, disgust, detestation seemed momentarily to hold a quivering conflict in the large pupil dilating under his ebon eyebrow." So she was right. So it probably tells us something about the relative abilities of scientists and artists to spot these things. Anyway, obviously these pupil size changes are of interest to people interested in cognitive science, psychology, neuroscience, and it's been known for a long, long time that there are changes in pupil size which seem to correspond with ongoing cognitive or emotional states, or cognitive operations or emotional states. These are relatively small, but they've been known about for a long time. It's only really recently that people have realized, in a sense, that they have data, if they've been doing eye tracking studies, that can get at this. Doing pupillometry 30 years ago was a far more effortful process than it is now. But because of these changes, the pupil has been described as a permanently implanted electrode, or the only visible part of the brain. And it does provide a potential physiological reporter variable that will allow researchers to independently measure psychological processes. So independent of subjective report. We're not asking people to say yes or no, make an old news judgment or anything like that. We can actually see this thing happen outside, in many senses, of their conscious control. So there's a whole huge literature out there that's not just recent on pupillometry, and it was started in many ways, or pioneered, I guess, by a person called Hess, who's a fascinating person, and I'd love to do a whole webinar on him at some point. But he published many, many studies showing that pupil size essentially changed with emotional valence, your emotional state. And his sort of central argument was that the pupil dilates in kind of positive emotional states and constricts in negative emotional states. As we'll see, this was probably not true. He published a very, very popular at the time book, as a kind of popular science book called The Telltale Eye, How Your Eyes Reveal Hidden Thoughts and Emotions. And he even did some very kind of modern sounding experiments. You can see here he's kind of photoshopped, or whatever the 1970s version of Photoshop was, these images of a model, so that here she has very dilated pupils and here she has very constricted pupils. And he was able to explore many things using this technique. However, and this is possibly why pupillometry kind of suffered a decline after Hess, his research was, at the time, very comprehensively discredited, most notably by Janice and Lowenfeld. And this is a quote, I think, from Janice, who says he was criticized for repeatedly using very small sample sizes, not reporting all potentially relevant results, not using evidence. I mean, it's a kind of absolute catalogue of all of the things you shouldn't do. And somehow, yet, he managed to publish in some quite prestigious journals an awful lot of papers. As ever in science, the kind of most damning finding, if you like, was the fact that many of his effects could not be replicated, particularly this idea of aversion restriction, the idea of your pupil constricting to emotionally negative stimuli or events. But it led to this very sort of widely received and understood idea that the pupil, which had presumably been understood for a long time before this, that the pupil reflects to some extent some concept such as arousal. Now, arousal is one of these terms, there are many in psychology, which can mean lots of different things to different people. But in the kind of pupillometry literature, it was often used to kind of, well, the manipulations used were things like explicit stimuli, fear of shock, things like that. And the idea would be that you'd increase arousal and demonstrate some kind of increase, corresponding increase in pupil size. And this has been replicated widely. There are hundreds of studies that show that this effect is real. If you increase somebody's emotional state one way or another, their pupils will dilate. But I think one of the problems with Hess's research was it kind of gave pupillometry a bit of a bad name for a while, which is a shame, because at the same time, Daniel Kahneman, who's far more famous for prospect theory and all of the other things, was doing some very, very interesting and very, very well designed and well controlled pupillometry research, looking at the impact of what you could call cognitive effort on pupil size. And he performed many experiments showing that a whole range of cognitive operations would result in pupil size increasing. For example, if you were trying to remember a sort of short-term digit span type task, as you loaded digits in, pupil size would increase accordingly. So it would be higher if you were trying to remember 4 digits than if you were remembering 2 digits, even higher if you were trying to remember 6 digits compared to 4 digits, and eventually when you exceed your span and you can no longer remember anything, pupil size would shrink back down again. And Kahneman proposed that arousal can be seen in a more general sense as a kind of response to task demands, and mental effort is part of that whole system, basically. So in a sense, he's arguing that we're measuring the same thing. So I want to touch now very briefly on some very basic physiology, which is taught at primary schools in the UK, and I'm sure it's known to you all as well. The main point I want to make is you have two muscles, or two main sets of muscles: the sphincter pupillae, which is involved in making the pupil size smaller, and the dilator pupillae, which are these radial muscles which are involved in making the pupil bigger. The only reason I'm making this point is to point out that both of these muscles are what's known as smooth muscles. The fibers within these muscles are smooth muscle fibers. This has huge implications for our understanding of what we're seeing when we're looking at pupil responses, because smooth muscle fibers are slow. There are actually three main types of muscle fiber in the body. There are skeletal muscles, cardiac muscles is the second one, and then smooth muscles. Skeletal muscle fibers can be fast. For example, the muscle fibers that actually move the eye, the extraocular muscles, are fast. But smooth muscle is found typically in the walls of hollow organs. These are blood vessels and gut, and they're responsible for the kind of dilation of blood vessels that you get to increase oxygenation, and the peristalsis and things like that in the gut. They are also the same fibers are in the pupil muscles, and this means that any change you get in a pupil is going to be a slow change. And I think in the next slide, or very slow change, in the next slide I just grabbed a graph from a paper showing a kind of standard, if you like, cognitively evoked pupillary response, which occurs here at time 0. And you can see that these authors are getting their effect, the kind of maximum dilation and then the maximum separation across their conditions, over 1,200 milliseconds after their event. Pupil size changes are slow. Okay, so if you're seeing fast changes, beware, be concerned. It's almost certainly not a result of any muscular change within the pupil itself. Now, another point to make about the basic physiology, because it has, again, implications for how we look at the data, is the fact that pupils do not dilate concentrically around their center. In fact, they don't even dilate particularly symmetrically or evenly. But it would be nice if, you know, here was a small pupil and here was a big pupil. You can see that for small and the big pupil, the center is still in the center. But in fact, what happens is something more like this. We have a small pupil with its center here. As it dilates asymmetrically, and of course that its center therefore shifts. This has consequences which I'll mention now for gaze data. So not pupil data, but the gaze data, the X and Y location reported by the eye tracker as to where you are looking. And sometimes people, for example, if they calibrate with a white dot on a black background, and then their targets are a red target on a white background, you have a very, very large change in luminance between when you calibrated, which would have been to a relatively small pupil, to when you show your stimuli, which will be a comparatively larger pupil because the luminance levels will have gone down. And this can cause small, but not insignificant sometimes, offsets in your gaze data. So it's worth bearing that in mind and recalibrating as necessary. This is actually a colleague's real eye illustrating that exact effect. Here's him with a dilated pupil, and here we have a relatively contracted pupil. I think he's shone the torch from his mobile phone into his eye. And here we've just taken these two images, made this one a bit transparent, and overlaid them. And you can see this asymmetry. It's also an asymmetrical shape, interestingly. So it's not equally dilated around the small one. It's dilated upwards and inwards up here. And people have done research. Andreas published a nice paper exploring the impact of this on gaze. Now, these are very extreme changes because he was manipulating the pupil diameter in an extreme way. These are not, well, these are larger than the kinds of effects you might get just within an ordinary experiment if, for example, there were luminance changes across parts of your image. But you can see there's individual differences, but there is a relatively consistent sort of pattern for the errors here to be downwards and inwards, presumably because the pupil's dilating upwards and outwards. I can't remember if this is dark minus light or light minus dark. But the point is the same. Not only, well, changes in pupil area can have a small impact on reported gaze. The last points I want to make on basic physiology are individual differences. So people will come into an experiment with very, very different baseline pupil sizes. And the implications for this, as we will discuss later on, are that you don't really need or want to be reporting absolute pupil values. Ultimately, what you want to be reporting is change, because there's so much change not only in baseline pupil sizes, it's also changes in how reactive people's pupils are. So if you shine a light in somebody's eye, the time it takes for the pupil to begin to constrict and the time it takes to constrict will be different across individuals. Not hugely, but there are individual differences. And then there are lots of other factors that can have an impact on pupil size at baseline and its ability to change. For example, age. As people get older, their pupils become less reactive, slower to change. Caffeine, nicotine, and in student samples, sometimes other pharmacological things being on board can all have an impact in pupil size. Even, I think, time of day, so when you do your testing. Again, the implications being you should probably at least attempt to control for these things. But the key point I want to make is that within an experiment, it makes sense to measure change rather than some kind of absolute area. Now, many journals, if you submit a paper that's reporting pupillometry data to a journal, they will ask you to report your pupillometry data in millimeters. That's reasonable. It provides a standard measure that works across eye trackers, for example, but you will probably be reporting change as a percentage of those millimeter values rather than absolute values. Again, some basic physiology. That constriction is under parasympathetic control, dilation under sympathetic control. The key point here being that these two things work together, so you can impact on either of these sides and have an effect on both constriction or dilation, in a sense. Pharmacology, again, I don't want to dwell on this. The key point here is that there are links to all sorts of interesting neurotransmitter systems that are interesting for psychologists in various reasons. So you've got mainly acetylcholine working for the parasympathetic pathway, the constriction, and norepinephrine or noradrenaline for the dilator pathway. And it's therefore unsurprising that various compounds can have an impact on pupil size. I want to talk now about the non-cognitive influences on pupil size, because these are things that it's important to take into account when you're designing an experiment. The most obvious one is the pupillary light reflex, which is obvious. When you shine a light into an eye, the pupil will constrict. When it becomes dark, the pupil will dilate. These are amongst the fastest changes in pupil size you can get. In fact, the pupillary light response, if you shine a light into somebody's eye, that reduction is about as quick as things get when the pupil size changes. So it's been shown it will reduce by more than 50% in just 200 milliseconds. There's a a latency to this, as we will see. It doesn't happen immediately. There's a reaction time, if you like, before the pupil begins to constrict. But when it does, it constricts relatively fast. Now, perhaps more worrying or more problematic for pupillometry research using eye trackers as recording devices is the lid closure or blink reflex. There's actually surprisingly little written about the effect of blinks on pupils, but imagine even if we weren't recording data with an eye tracker, there is clearly, if you blink, it becomes dark, there is an effect. It's not always clear exactly what this effect is. The effect of the eye tracking data, as the eyelid descends over the pupil, it will result in an ever smaller portion of pupil left for the eye tracker to track. So the eye tracker will see what looks to it like a very, very, quick reduction in pupil area, and you will see this in your data. Eventually there will be so little of the pupil left that the eye tracker can no longer compute its center, and the eye tracker will then signal a blink. But that point will be reached after 10, 20 samples, during which the eyelid has been descending over the pupil, resulting in a rapidly decreasing pupil area signal, and actually resulting in a shift in the downward center of the pupil, or the visible pupil, that is sufficiently fast, has a sufficiently high velocity, that it will actually trigger the saccade parser in the eye tracker. So you will find that all of your blinks are actually wrapped in what are, in many cases, artificial saccades. This is another thing we're going to look at. In some cases may actually be real saccades, because one of the things that the brain does is it kind of minimizes the downtime to vision, and we often combine saccades with blinks. So you may stare at a single dot in the middle of the screen and blink and still be staring at it at the end, in which case you'd have a blink wrapped in an artificial saccade. Or you may stare at a dot in the screen and then look at another dot, and at some point during that process you also blink. So these things again are things to watch out for, and we will look at when it comes to the data. But as you can see, there are clearly changes in pupil size that occur after blinks. I actually took a quick look at a whole random selection of data, just exported hundreds and hundreds of trials. I think there's over 1,000 blinks here, and just literally chopped out the blink, and this is data from an iLink, pupil area data, and you can see this is the point at which the blink was signaled, and the blink could have lasted 100 milliseconds or 200 milliseconds or longer, but I simply chopped the data off at the beginning of the blink and then pasted it back in from straight after the blink. And you can see that before the blink itself is actually signaled, there's this rapid drop that will go all the way down to 0. I rescaled the axis because otherwise you can't really see what happens afterwards. And eventually, as the eyelid begins to come back up, you get this rapid ascent of pupil area. And then this is the kind of pupillary response, if you like, after the blink. You can see this kind of slight dilation. This is relatively small given the overall scale of change that could occur. And then a kind of settling down, and then presumably, I'm not sure what was going on in this task, but there was a kind of ongoing increase in pupillary response after that. But as you can see, things don't really settle down until good 600 or 700 milliseconds after the end of the blink. So these things again are things to bear in mind when it comes to analysing your pupil data. It may be that your pupil analysis isn't looking at pupil size in this kind of fine-grained way, but if it is, you might want to consider certainly excluding this part of the pupillary, of the blink reflex, when the eyelid's descending and ascending over the pupil, maybe even possibly this. But I will come on to how we would do this in Data Viewer in a moment. There is another influence on pupil size, which is almost certainly not relevant, but I mention it just in case it is. But when you focus on things that are close to you, there is a response. Your pupil will constrict in order to keep things focused. Because most eye tracking studies are done with a screen which is at a fixed distance, this generally is not an issue. I'm mentioning it for completeness. Now this is a big one, the pupil foreshortening effect. Because in many eye tracking studies, it's not unreasonable that you may also be interested in gaze as well as pupil size. In other words, you want to know where people are looking, which implies that people have got things to look at, in which case they will rotate their eyes. Now, because the camera is fixed in its location, it has a fixed view of the eye, as the eye rotates away from the view of the camera, you can see that what was a nice, clear, circular pupil there becomes slightly more ellipsoid. It now has a taller x-axis than y-axis, if you like, and that will result in a change in what we are measuring, which is pupil area or diameter. It's just illustrated here with schematics rather than the eyes, but you can see the same thing. As the eye rotates away from the camera, it will cause a change in your pupil area measure. And we'll come on to how to deal with this, or ways around this, in a minute, but it is up to 5 to 10% of your pupil area measure may be influenced by where the eye is. In other words, if you took a completely static robotic pupil, an artificial pupil, and rotated it, you could get a change in your pupil size measure from the eye tracker up to 10%, even though clearly the pupil itself hasn't actually changed size. So this is also something to bear in mind. The last, well, there are other complications, one of which is this thing called Pupillary Hippus, which in many senses may actually be what we are trying to mention. It's simply the term for the fact that the pupil itself is never ever still. It's constantly dilating and constricting slightly. And even if you look really, really closely, people have shown that you can see dilations that are linked to cardiac rhythms. In other words, it's known that the iris itself is vascularized, and it may well be that just as these blood vessels throb, there are slight changes in the pupil size itself. The implications in terms of experimental design are that you need to baseline, and you need to take baseline pupil measures regularly so that you can report some change. And that baseline itself probably needs to be some sort of average over a reasonably long period of time, because otherwise you're just randomly sampling somewhere within this continually changing signal. Interestingly, there are interactions here between the extent to which your pupil will show these kind of constant changes and the levels of luminance. So it becomes more obvious in brighter light. That may simply be a kind of ceiling effect. If it's in dark light, it may be that your pupil is already so dilated that there isn't much more dilation it can do. These are other factors that we need to take into account. This one, I think, is possibly less known about, but if you do any pupillometry research yourself, you will run into this. This is just average pupil size across a whole trial for many participants over the course of the entire experiment. And you can see pupil size starts off in the first trial quite large and then descends very rapidly. Then there's a big spike here. You can see another big spike here. These spikes are the end of the block. So this was a little practice block of 12 trials. You see pupil size decreasing rapidly over those 12 trials. Then we have a little break, and then there's a screen that says, okay, and now the experiment's going to begin. And oops, up pupil size goes again, and again descends pretty rapidly over the next few trials, but then continues to get even smaller till the end of the block, where it bounces back, but not quite to the same levels as the first break, and again quickly descends and then shows this more gradual descent. Now there are all sorts of things that could be going on here. When people come into a lab at the beginning of a task, they're unsure of the environment. Maybe they've never seen an eye tracker before. They don't know what the task's going to involve. For example, all of these things will lead to an increase in arousal and in probably processing load. So it's unsurprising that at the start things are generally relatively dilated. Then as the task progresses, it becomes clear what the demands are, you probably get good at it, and cognitive demands drop off. These even longer term changes, after you've had that kind of initial drop off of, okay, I know what I'm doing, may reflect boredom, they may reflect fatigue, maybe further learning, who knows. But if you plot your pupil data over time like this, just sort of basic or average pupil data, you will see these effects. And again, it highlights the need for baselining within a trial, because if you just reported absolute values, you'd be getting confused by this compared to this. What you need to know is what changed within this trial. Okay, so task-evoked pupil size changes themselves are slow. They also have a relatively slow latency, beginning a few hundred milliseconds post-stimulus, peaking around 1 to 2 seconds post-stimulus. So again, this all has implications for how we analyze our data. We need to make sure we're exploring enough of it that we've fully grabbed the full response to whatever event we have in our experiment. It also has implications, I think, for things like reading research. It might be nice to think that, you know, we could use pupil size to see how difficult it is to read certain words, but you have to bear in mind the fact that we don't spend very long fixating individual words, and it may well be that the response you're looking for is going to happen after gaze has moved on to other words, for example. The key point here is that it's an indirect measure, and there are lots and lots of things that can impact on it. So there's a problem of psychophysiological inference, which is the assumption that there is some kind of consistent one-to-one relationship with whatever it is that we're exploring. And with pupil size, we know that's not necessarily the case. There are lots and lots of different things, for example, links that may impact on pupil size or where the eye is, all of which we need to take into account. And just as a last kind of piece of background, just to point out just how sensitive pupil sizes are to all sorts of things that might not be what you're interested in. This is showing the pupillary response into change in how hard you clench your fist, basically. And this is the pupillary response to somebody lifting weights, which is just little weights, lifting them off a desk, versus not lifting weights. So you can see that physical effort, not just cognitive effort, can impact on pupil size. So all of these things are impacting on pupil size. All of them need to be borne in mind when it comes to the analysis stage. So let's move on now to considerations for our experimental design. We know pupil size fluctuates continually, so we need to take baselines. We know that pupil size is readily influenced by many non-cognitive factors, such as luminance, such as where gaze is, so we need to take these into account. We know that there is a robust relationship with a somewhat slippery concept, mental effort, cognitive effort, which inevitably is going to be involved in most tasks. Anything you ask somebody to do is going to involve some mental effort. And we know that eye trackers were not designed as pupillometers. In other words, they do not, well, because people are allowed to move their eyes and because the camera of an eye tracker is essentially fixed, there are some issues there that we need to address. So let's address these issues. There are some general design considerations, perhaps the most important of which is that, in my opinion, it makes most sense if you're interested in doing pupillometry to use the head stabilized mode. In other words, you use a head support, and ideally you would keep your camera to head distance or camera to eye distance fixed for all of your subjects, which is relatively easy to do if you always make sure that you keep the top half of your head support in the same place and just adjust the chin rest up and down to suit your participant, and adjust the participant up and down in a chair to suit headrest. This also has the advantage of it makes sure that people's eyes are always aligned with the top quarter of the screen, which is what they should be according to our optimal setup guide. So if you keep the forehead bit there and put a face in here, the eyes are going to be the same distance from the camera, assuming that you don't move your camera, for each participant. As we will see, this is important for when it comes to converting pupil size to millimeters. Remote mode is not advised, mainly because in remote mode people are free to move their heads, and they could easily move their heads closer, further away from the camera. Now obviously this would make the pupil appear larger or smaller on the camera screen, which would impact on your pupil area measure. But also what happens is as you go further away from the camera, the illumination, the amount of infrared reaching your eye is less, the eye becomes darker, so maybe more pixels on the pupil are thresholded. Or as you become closer to the camera illuminator, everything gets brighter, maybe fewer pixels are thresholded. It will, at the very least, add an awful lot of potential variability to your data. There are things you can do. You can limit these kinds of forwards and backward movements while still allowing other head movements by encouraging people to rest back into a high-backed chair, or if you're working with infants, you can use the kind of harnesses you get in a car seat, which will stop them rocking forwards and backwards, for example. If you have an iLink 1000 Plus, then I would thoroughly recommend that you use the 25mm lens if you're going to try this in remote mode. It does a closer zoom on the eye. You will have more of the camera pixels covered by a pupil, and you will get less noise in your pupil data and more movement in it, to be frank. So use head stabilized mode. Try and keep the camera distance fixed for all participants, as I've said. Now, some people have asked, well, look, if all I'm interested in is pupil size, surely I don't need to bother with calibrating. As we'll see when we come on to dealing with the pupil foreshortening effect, it can be very useful if you have accurate gaze data as well, or alongside your pupil size data, so that you can take it into account somehow and make sure that this pupil foreshortening effect isn't somehow confounding your data. Now obviously, because pupil size as reported by the eye tracker is simply a reflection of how many of those little pixels on the camera retina, or the camera sensor if you like, are thresholded, in other words are coloured dark blue during recording, then if you adjust these pupil thresholds during a recording, you are going to artificially change your pupil area measure. So once you've got them set up at the beginning, or at the beginning of each block, or each time you do camera setup, then keep them like that for that recording, so that any measures of change within your recording are going to be consistent. The iLink systems give you the choice of whether you record area or diameter. My personal preference is for area. Diameter is simply derived from the area, so it kind of doesn't matter which one you choose. It's also then scaled so that it has a similar granularity as area, so it really doesn't matter which one you choose. I tend to stick with area, but what is important is you try to be consistent, so you don't end up recording data from half your participants with area and a few with diameter, because then you're going to have to convert everything back so that they're in the same scale, which I will show you again how to do towards the end of this webinar. So those are sort of general setup issues. How do we control luminance? Well, there are two sources of luminance changes that we need to control. One is the ambient illumination, for example, how much light there is in the room, and here it's obvious advice really, try not to do your recording in a room that has large windows that don't have heavy curtains on it. For example, in the UK at least, where it can be very, very dark when there are clouds and then all of a sudden the sun comes out, if you're in a room with a window that isn't shielded very well, then this will have an impact on pupil size. So normal artificial lighting and in a window if you have got windows in your lab, if they've got decent blinds or decent shutters, should be absolutely fine. A slightly more tricky issue is what I call local luminance changes. So if you were looking at a kind of an image on the screen which the top half of the image was black and the bottom half of the image was white, your pupil would be more dilated when you were looking at the black half than when you look at the white half. So these kind of local luminance differences can have problems, and were indeed problematic. Many of Hess's original research was criticized for not controlling these kinds of things. So there are various things you can do. One is to try and use isoluminant stimuli. So for example, words are great stimuli, especially if they're from the same length, because they're generally going to be isoluminant. Now clearly this is not a strategy that's going to work for many types of tasks. Maybe if you're looking at face processing or something, you can make sure that all of your faces are broadly the same luminance levels, and you can control with this in software. For example, you can retint images or rescale images using various image processing software. But this isn't always possible, so really the simplest thing to do is to try and make sure that there's no systematic confound between luminance and any of your experimental manipulations. And I put remember Hess there because Hess, for example, showed arousing images or positive valence images, which were generally images of naked people, so they were quite kind of light, and then, or the other way, I can't remember. Anyway, there was a confound between the types of images he was showing and their luminance, basically. So if you have got a load of stimuli, you can very, very easily load them in, I think, into MATLAB, and it will just spit out the kind of overall brightness level of the, you know, the colors in all of the pixels, basically. You can use that as a very rough check to make sure that you've got no kind of gross luminance confounds. So it's not the case that all of your stimuli in the happy condition are brighter than all of the stimuli in the sad condition, because if they were, that would clearly lead you falsely to conclude that people were more aroused to sad faces because they were actually darker, not because they were sad. Shut things out, natural light. Some people think they should go to sort of great lengths to determine the midpoint of the pupil range and then sort of adjust some ambient light level so that for the entire task, or at least at the outset of the task, pupil size is somewhere in the midpoint. I really don't think that's possible. That's all that sensible. Mainly because of those long-term changes that I outlined in an earlier slide. If you baseline it at point A, that's absolutely no guarantee that in 20 minutes time you're going to have the same baseline. For most people, normal daytime light levels, whether it's artificial light or natural light, will have their pupils somewhere roughly in the midpoint of their pupillary range, which means you're going to be able to see constrictions, you're going to be able to see dilations. Obviously what you don't want to do is do everything in the dark, for example, and then hope to see some cognitive evoked dilation, because pupils can only dilate so far. If they're already dilated as far as they can because it's dark, you're not going to see anything. Similarly, if there's a really, really bright light source and the pupils are constricted, it may limit the amount to which they are prepared to dilate due to cognitive influences. So normal natural light, somewhere in the beginning, in the middle, is perfectly fine in my opinion. How do you get around the fact that the pupil size is continually changing? Well, as I mentioned before, the basic idea is that within each trial, you want some point at the start of your trial where you're just going to record pupil size for maybe 500, 1,000 milliseconds. Now, importantly, and you'll see that this is important when we come to analyze the sample data set I'm going to show you, ideally there would be some isoluminant stimuli on the screen at that point. So you wouldn't just have people fixating a white target cross in the middle of the screen, which then changes to a face after 500 or 1,000 milliseconds, because clearly when the face appears, as we will see in the data, this is going to have a large impact on pupil size. What you want is some kind of isoluminant stimuli. So when I used to work with words, it was very, very easy. You could just have 5 letters X on the screen, and then after 1,000 milliseconds, each one of those would change to another letter and the word would be revealed. There's no sort of concomitant change in overall luminance levels, so any change that you observe in the pupil after that event must be cognitive. So isoluminant baseline stimuli are very, very easy. And I will show you in a minute, there is again MATLAB scripts that will let you import image files and scramble the pixels basically. And this is a very, very sort of sensible thing to do in my view. But this really is the key idea. Everything that you report and record is going to be pupil size subtracted from whatever that baseline time was, or whatever pupil size was, the average pupil size during that baseline. Because then it doesn't matter what people's overall levels of arousal are, it doesn't matter where they were in their sort of Hippus thing, you've basically controlled for and are measuring change within that trial, change to specific cognitive events. How do we control for blinks? Well, this is much harder in many ways. Obviously, you could keep trials short, trying to discourage blinking. So if you've only got trials that are 2 or 3 seconds long, maybe a bit longer, you could have a blink screen, as you would, for example, in an EEG experiment, during which you encourage people to get their blinking out of the way and prepare themselves for the next trial, and they will make an effort to try and keep their eyes open, or not to blink during that trial. I've generally not gone down that route, partly because I find that if you mention blinking to people, blinking becomes an issue, and they will either try not to blink, in which case their eyes will start watering, they'll be forced to blink, or they will blink excessively or something like that. The other alternative is excluding trials with blinks. Again, that only really works if you have relatively short trials. Any trial that's going to be longer than 6, 10, 15 seconds is almost inevitably going to contain a blink at some point. So how do you get around that? The only solution, if you have long trials and you're looking at pupillometry or pupil size changes at some point over a longer time course, is to remove the pupil data during the blink and probably a few samples, or quite a lot of samples, before and after the blink, and then either interpolate or fill. So if you interpolate, you just take the pupil size from before the blink and do some kind of linear interpolation up to whatever the pupil size was after the blink, or you can fill, so you could just fill that gap with the average pupil size data, for example. I'm not hugely keen on this, mainly because in my research I've generally been looking for a specific response, which I know roughly when it's going to come, and if I've got enough trials I can then get rid of any trial in which there happened to be a blink within that 2,000 millisecond window that I know that I'm interested in. You tend not to lose too much data in that way. But again, I will cover in a bit how you might go around or go about doing these kinds of corrections, if you like. Here's the MATLAB code. I found it on the web. It's completely untested. I'm just leaving it in this PowerPoint file, which I will put up to our support forums in case other people use it. But basically, you can read in a JPEG, set some kind of block size. So how many, what size of block do you want to then shuffle, and then shuffle those blocks, and then write out the scrambled image at the end. And you could use that to create isoluminant baselines, assuming that you've got some kind of pictorial stimuli that you're working with. How do you control for the pupil foreshortening effect? As I've said, this can impact pupil area data or pupil size data by up to 10% at its most extreme. Generally, it's smaller than that. People have published interesting research sort of going along the idea that if you take an artificial eye, or even a real eye, you can measure what the effect is on reported pupil and then create regression equations and use those to kind of regress out, if you like, the foreshortening effect's impact on your data. I think one of the sort of my slight concerns with these kinds of approaches is that the pupil is continually changing size and shape, and it may well be that a kind of regression function that makes sense for a 4mm pupil may not be quite so accurate if the pupil is in fact 6mm, and it may well be that as the eye moved from one point of the screen to another point of the screen, there is both an artificial change in pupil size due to the foreshortening effect, but there's also potentially a real change in pupil size if pupil size was changing over that time, in which case it becomes difficult to kind of disentangle those. So my preferred approach has always been to use relatively small central stimuli, which will minimize the kinds of eye movements that people make during the task, and therefore minimize any impact of the pupil foreshortening effect. Clearly, for many people, if you're looking at natural scene viewing or people viewing works of art or something like that, then this isn't possible. You know, you can't stop people or restrict eye movements. And this is why, as I said before, it's important that you have calibrated data, because for my money, by far the simplest thing you can do is to throw the gaze and x and y coordinates into your analysis as covariates, and then you can see whether any effect that you've got on pupil size is being confounded by some kind of pupil foreshortening effect. In other words, if people look at happy faces and sad faces, and you find that pupil size is bigger for happy faces than sad faces, if you throw the x and y coordinates of your maximum pupil size data point in as covariates, you'll be able to see, is this because it just so happens that when people are looking at happy faces, they're always looking at the top, whereas when people are looking at sad faces, they're always looking at the bottom of the face. So hopefully you don't get an effective covariate. If you do, then you need to investigate whether you've got something, potential confound there, that may be due to the pupil foreshortening effect. The other obvious thing to do is to make sure that there's no systematic confound between your experimental manipulation and gaze location. In other words, or make sure you balance your stimuli location. You haven't always got the target appearing on the left, it appears equally often on the left or the right. Or if you've got some visual search task, the target appears equally often and the distractors appear equally often in each of the potential target locations. So, as I say, this is a kind of example of kinds of things I've done, and you'll see when we come onto the data. But balancing across conditions, including as a covariate, these are the ways to go. Now, some eye trackers do report pupil size in millimeters. Our systems do not. We report area or diameter in arbitrary units. And as I said, these arbitrary units are simply a reflection of the number of pixels on the camera sensor that are thresholded as the pupil, in other words, that are covered in blue. So if we want to report millimeter data, and as I mentioned before, many journals will want you to report millimeter data, then we need to do something about it. And the solution is basically to print out an artificial pupil with a known diameter. As an example here, I said 6 millimeters. Now, what matters isn't that it's 6 millimeters. When I first did this myself, I spent ages in PowerPoint trying to print out all sorts of things until I got a circle that was exactly 6 millimeters. All that matters is that you print out a circle somewhere within the range of, say, 3 to 8 millimeters, and you know its measurement. In other words, once it's printed, you can measure its diameter, and then you know that diameter. And then you literally place this artificial position in your head support at the same position roughly as somebody's eyes would be, with your camera at the same distance that it was for all of your recordings. This is why it's important that you keep that camera to eye distance the same, because you don't want to have to do this for every single participant. So you put the artificial pupil that you've printed out into the head support, and you point the camera at it and you record some data. The idea is you see what area or diameter values you're getting for that artificial, and then you simply create some conversion factor based on the average of the data that you recorded to the artificial pupil. I'll go through this in a bit more detail. First, if you're going to try to get our eye tracker to record a printed black circle, you're going to have to tell it not to look for a corneal reflection because it's not going to find one. And it looks a bit complicated, but again, this is here simply so that you can cut and paste from this PowerPoint. You paste these lines into final INI, and you then relaunch the eye tracker. So final INI is the INI file that's read in last of all of the INI files in the host software directory. It will override any of these settings that have been set previously, and having done that, you will find that when you go to record your data, you can select pupil only mode on your eye tracker. So you can select pupil only mode, you can then track your artificial pupil. Don't forget to remove these lines from final INI before you go off and collect actual data on participants. So one thing I've seen people do is print out and record a range of different artificial pupils. It turns out if you do that, and I've done this myself, the correlation or the, is sort of very, very strong. So you get a very, very straight line. So really you actually only need to print out one of any single size. So a single target will do. So let's say we've printed out an artificial pupil size, an artificial pupil of 6 millimeters, and we record it in our pupil only mode. We open up that data, output the pupil data, so we'll show you how to do, and we see that on average we're getting an area value of 1000. So if that's the case, we know that in our actual data a pupil area value of 800 would equate to 5.37, because you can literally calculate how many if you like, millimeters each one of these camera sensor or area units would equate to, and do the maths, basically. So the simple maths is whatever your pupil data is, take the square root of it, divide it by your scaling factor, and multiply it by your scaling factor, and you will get the new value in millimeters. If you're recording diameter, then you don't need to square the pupil data. The squaring here is simply because an area is an area, and we want to kind of linearize things to a single dimension, so we take the square root rather than so if you're just working with diameter, then you can just omit the square root from these equations. And that's how you convert to millimeters. It sounds relatively straightforward. It's a bit of a chore because you have to paste that data into final INI, paste those lines into final INI, record a bit of data with an artificial pupil. But once you've done it, assuming that for the rest of that experiment, all of your participants are tracked at the same, with the camera at the same distance, then all of your millimeter values will be meaningful. Now, if, as can occur, you find out after collecting all of your data that somebody had used the eye tracker whilst you were using it and had set things back to diameter or from area to diameter or whatever, and you've accidentally got some people whose data is recorded with pupil area and for others it's pupil diameter, then you can use these two little conversion equations to go from one to the other. As I said, diameter is derived purely from area and scaled, and these equations will show you how to do that if that ever occurs. So finally, and about time, I'm going to go through some actual data analysis itself, just very, very quickly. For those of you that have seen the webinar on using Data Viewer, it's essentially the same data set because it happens to be mine and it happens to be easily available. So just to give people a quick refresher of what the experiment was, people stared at faces or fractals for 4 seconds. Simple as that. Faces were either neutral faces, fearful faces, angry faces, or happy faces. And the experiment itself was nothing to do with pupillometry. We were interested in asymmetries of facial processing. So we're interested in which eye did people look at first? Did they spend more time on the left or the right side of the face? Does this correlate with their handedness, with schizotypy scores, all sorts of things like that. Pupillometry and pupil size was absolutely not in our minds when we collected this data. It just happens to be data that we have, and as we will see, it happens to be, so happens that there's some interesting pupil effects. But you could, for example, make some predictions at this point. You might predict that pupil size would be greater to fearful and angry faces, these kind of more negative emotions, than to neutral faces. Or you may predict that pupil size would be larger to all faces compared to fractals, because fractals, or maybe emotional faces compared to fractals, because fractals aren't delivering any information that requires processing or responding to or worrying about or anything like that. So that was the experiment. 5 levels of condition really: neutral, fearful, angry, happy, and fractals. And as I said, the experiment was not designed with pupillometry in mind, but luckily, or usefully, before these faces were presented, there was a random fixation time of between 500 and 1,000 milliseconds, where people simply looked at a cross, so it's not isoluminant, but we can use that to get some kind of within trial baseline that we can then use for our pupil data analysis. So at this point, I'm going to flip out of PowerPoint. I'm going to leave this PowerPoint up so that I can remind myself. These really are just reminders for myself as to what we should do. And I'm going to show you, just really walk you through in the next, I guess, 20-30 minutes, an exploration, if you like, of pupil-sized data that allows us to see how you might want to factor in some of these considerations that I've talked about, things like blinks, pupil foreshortening effect, and things like that. Okay, so, and I will again upload this data set to the website so that people can explore it in their own time. So hopefully here you can see the data. We have, I think, about 20 participants. It's a sort of subset of a much larger data set actually. But you can see I was fairly cruel back in the day with my PhD students, and there are 240 trials in this task, and each trial is a face, could be black or white or color. That's another manipulation that I'm not going to look at in this particular analysis, or it could be a fractal like this. So, and you can see that whilst gaze is generally relatively restricted, these aren't full screen stimuli by any means, they're relatively small and central, and therefore any impact of the pupil foreshortening effect is going to be very, very minimal, given these sorts of movements around the face itself. You do occasionally get people glancing off to the side and things like this, and this is something that we may want to take into account, because, you know, if glancing off to the side happens to artificially result in a dramatic increase or decrease in pupil size, we might want to know that that's what's happened in this case. So if we play back a trial, you will see that at the beginning there's that fixation cross, and then the face appears. And the first thing we want to do is to define 2 interest periods. And if we look at our data in the sort of time view format, the 2D temporal graph mode, click on the little message button, we can see when we've got messages coming in. And here you can see this is my display stimuli message, and this must be my display fixation message. Now, we know that this period here is random. It could be anything between 500 to 1,000 milliseconds. We want to make sure that we've got 500 milliseconds to do our baselining from. So really what I want to do is define an interest period that takes this message at its start point and goes backwards in time, if you like, 500 milliseconds, because I know that there's always going to be 500, at least 500 milliseconds from this point. So that is what I will do. And in fact, I've actually done it. But if we go to look at how we do it, I put in a label for it. So I call it fix less 500. So it's 500 milliseconds. I suppose it should be really stim less 500, shouldn't it? The start message is display stim, but the offset I put in here is minus 500. In other words, we want to start this interest period 500 milliseconds before this message appears, whenever it appears. And then I've made it a duration-based interest period, and I've said its duration should be 500 milliseconds. So this interest period is going to give us the first 500 milliseconds of our data. And if we go back to the overlay view, you can see now that all I can see is a cross. So there's our baseline interest period. We also want to make a period, or an interest period, a window, which covers, in this case, I've just done the first 2000 milliseconds because I'm expecting that within 2 seconds of this target or this face or fractal appearing, any kind of pupillary response will have had time to reach its maximum. So if it is an angry face, within 2 seconds you should have had enough time to realize that it's an angry face, and anything that's going to change between it being an angry face or a happy face or a fractal will have happened within 2000 milliseconds. So here again I have display stim as the start message, no offset this time because I want to count forwards from this point, and again it's a simple duration based interest period, and I've set the duration to 2000 milliseconds. So we've set, and if I set that interest period, you can see we've now got a face. So the first thing we want to do is to derive our baseline. So let's choose the baseline interest period, just that 500 milliseconds when the cross was on the screen, and let's run a report. And for this baselining exercise, where all we want to do is find out what the average pupil size was within this subset of the data within this interest period, then the trial report is very, very useful. And you will see I've chosen a few variables. These are variables that are part of my experimental script. So block is the variable that actually encodes condition, believe it or not, whether it was a fractal, happy, sad, whatever. Image is the image name. This is just trial. I think type is whether it was color or black or white. I've output the interest period start time. This is important because one of the things that I've output is the pupil size max time. In other words, at what point in the trial did pupil size reach its maximum? That's going to be a sort of measured in tracker time, as is the IP start time, so we can subtract one from the other to work out how long into the trial itself did it take before pupil size to reach its max. But I basically output here, I've chosen all of the pupil size measures that Data Viewer will give you. The maximum pupil size for that trial, the mean, the average pupil size for that trial, the minimum, the time at which the pupil size reached its maximum. This is the important one, the X and Y location at its maximum, so we can use this potentially to exclude outlying values which may have been driven by large shifts in gaze. And the same for the minimum, the time at which the minimum pupil size was reached within that trial, and the X and Y gaze location of the eye within that trial. And we can click next. I've already done this because it takes quite a while for this computer to save this session, but if you do run the report, we can then open that report, and it's this one here. Excel often moans when you open reports from Data Viewer because even though it gives, Data Viewer can give the reports an XLS extension, they're not actually XLS files at this point. They are simply text files, tab delimited files. Actually, let's run it because I can see here that this report doesn't have all of the data in it. Hold on 1 second. So analysis, report, trial report, next, save the changes. Okay, so I'm going to call this, well, I'm going to call it that one, we'll overwrite it. Data viewer churns through all of the trials, 240 for each person, and outputs that data. And this is really, if you like, the beginning of our pupil analysis. Once we've got that baseline, then we can start using it in useful ways. Whilst that's going, I'll just go back. We're going to open it in Excel, and I'm going to show you a trick again that if you watch the Data Viewer webinar you'll be familiar with, which is using pivot tables in Excel. You can use similar tricks in R or MATLAB or SPSS or whatever your program of choice is, if you like. I'm kind of fairly low level, so I like to use Excel because it's easy. So I use pivot tables in Excel. I think Data View is finished, so we can go to the output folder of our analysis, and there is the file we've created. If we open this in Excel, sorry, we can now simply highlight all of the rows, Control-Shift, right and down will highlight everything, and click Insert Pivot Table. I generally choose to have the pivot tables just somewhere over to the right of the data. And now we can construct a pivot table, and we can use it to explore, for example, our average pupil size as a function of our condition, fear, factor, happy, neutral, or angry. Now, at the moment, this is interesting, you can tell I've made a mistake in my experiment. These should have been mostly balanced, but we've actually got an extra angry trial instead of a fear trial. That's interesting. Anyway, that's because when you create a pivot table, the values default to a count. So this is just telling me how many trials of each type I've got. I've got 49 angry trials for that person. We don't want count, of course, we want average, and you can change that very simply by clicking on the little chevron there, clicking on value field settings and changing it to average. And if we do that, you can see quite encouragingly, I think, that the baseline pupil size across conditions is very, very similar. It's not really a huge difference between any of those. So that's encouraging. However, we might just want to check, you know, are any of these averages taking in data that happens to come from a point at which somebody blinked within that baselining session. So we could, for example, quickly look at blink count, and you can see that there are in fact blinks within that time. Not often, but some. So we may want to then use a filter. So we put blinks in as a filter, and we can say, well, let's only have our averages coming from trials where pupil size mean, and we can see we've still got decent numbers of trials there, where the blink size is 0. And again, we can see these values slightly more variable, but still broadly very, very similar and equivalent. Now, one of the things that we can do in Data Viewer, in the preferences, is to choose, in this data filters section here, the duration of samples or data to exclude before and after blinks. And you can see here I've put in 100 milliseconds for each. This is entirely arbitrary. If you think back to when I was talking the PowerPoint slide about the change in pupil size that followed a blink, you saw that this didn't really resolve for 600-700 milliseconds. But because I've only got a 500 millisecond window here, I'm not going to exclude too much. I just want to make sure I've excluded those very, very sharp drops that we saw at the beginning and at the end of blinks, because they will have very, very low pupil values in that I don't want. So already the values that I'm getting in my report are not including pupil area values that come from within 100 milliseconds, basically, of the start or end of a blink. How much or how little you do of this is unfortunately entirely a decision for you. But for me, 100 milliseconds makes sense. It's definitely long enough to get rid of any of the eyelid descending artifacts, basically. So we've run our baseline report. The next job is to do, I think, the simplest possible pupil size analysis you can imagine, is to do exactly the same thing, but now for our 2000 milliseconds. We'll run a trial report. In other words, we're only going to get one row per trial per participant, but we will get minimum size, max size, all of those things in our data, and we can then start to see how things have changed. So again, all we have to do is change our interest period. We're now going to export data relevant to the first 2000 milliseconds after the face appeared, and we can simply run the same report. Use the same variables. Data viewer will save itself again, and then it will run that report. And we can then go and see, well, let's just take, for example, average or maximum pupil size during that 2000 millisecond window. Is there any effect? What about confounds? What about blink counts? What about where the eye is at the time to max? Things like this. All of this we can then explore in that data. Data view is nearly finished. So we would call this our STIM 2000, STIM plus 2000 trial report. We've already done it, but I'm going to overwrite it now. And at this point, we can start to do some more interesting things. So where is it? STIM 2000 trial report. That's the one. If we open this in Excel, Excel will moan. We can do the same thing again. Select all the data. You can see we have the same number of rows as before. Insert a pivot table somewhere over here on the right. Made a mistake. Insert pivot table on the existing website. Okay, so again, we can put in our participants as rows, our block, condition if you like, as columns, and we can start exploring. Well, one thing we could do, for example, is create a new column called mean diff, and another column called baseline mean, and we could go to our baseline data, get the pupil size mean, paste that in here and simply calculate a difference between our pupil size mean during the task and pupil size at baseline. Mean. So equals this minus this. And immediately we can see something slightly interesting, which is that most of these values are negative. In other words, the pupil has got much smaller from the baseline to when the faces were on the screen. Now this makes total sense. When the baseline was being taken, there's nothing but a tiny white cross on the screen. When the faces or fractals appear, all of a sudden luminance increases massively and you get a massive drop in pupil size. And we'll see this in real time, if you like, when we plot the pupil size. But it's a very good example of why having an isoluminant baseline is important. But let's for the moment take our pupil size mean, don't want the count, we want the average, and we can see what looks like differences. So if we take this data equals U25, and we can copy our labels. Don't need the total one. We can plot quickly, this is why, and we can see that there is a clear tendency in this uncorrected data for pupil size to be largest for angry faces, pretty equivalent for fear, happy and neutral, and smallest for fractal faces, which looks like an interesting, possibly predictable effect. It's interesting that it's angry, not fear, or that you get it more for angry, not fear. So already, just very, very quickly, we've taken a kind of very, very rough and ready look at our pupil data. We could be better and look at the difference data. If you add columns to a pivot table like I have, and they're not in the original, you can click to the design, to the data source, and just expand it out so that it includes them. So now we could look at the mean difference. Don't want count, we want average. It doesn't like that. And change it to if error and if we can't subtract it, then we'll just take I2. So that if error function is quite useful. So basically I subtracted it if there was something to subtract from. If not, I just gave it the other one. So now you can see in our difference data, it's kind of the other way around. Things are getting less small when the angry faces appear compared to the fractals, and the other three are somewhere in the middle. So the same effect essentially, but now at least we've kind of allowed for these inter-trial baselines. Now, what happens if we exclude, use our blink count variable to filter? So we can see that there may be many blinks. So let's just look at the trials where there were 0 blinks. Has that had a big effect? It has. And one of the things, if you remember from this previous data, this is the nice thing about playing around. If we just put in blink count and change that back to count, and obviously change this back to all, one of the things that we found in this original data, which is entirely unexpected, is that there is a very, very noticeable reduction in blinks to, no, let's not count, let's give us some, sorry. There was a reduction in blinks to faces compared to fractals. So there is all sorts of potential confounds here. And we could either go to our raw data, or we could exclude trials that have got blinks in, or we could try and account for it somehow at the analysis stage by using covariates. One of the things that we can do is explore at what point is this maximum being reached. Whoops, average of, so pupil size max time itself isn't meaningful because it's in tracker time. As I said, we need to subtract it from the interest period start time. So we need to make another little column called max time, which equals this minus this. There we go. And now hopefully if we tell Excel about the fact that our data source is bigger, it should be in there, max time. Oh, I just need to refresh it. There it is. Don't want count. You can now see the point at which pupil size is reaching its maximum after that. And again, check whether there's any differences. And it does seem to be, which we will then go and explore. So the next thing I want to show you in the data is how we actually look at pupil change over time. So this is one very, very quick and simple technique to look at various things to see whether there's any kind of obvious effect on maximum pupil size minus during the sort of window of interest minus the baseline average pupil size during the baseline period. You can do that very, very quickly, very, very simply with a trial report. But I also want to show you something in DataViewer which can be very, very useful. And that's, it allows you quite simply to kind of get at a much more fine-grained analysis of your pupil data without having to look at pupil data at the sample by sample level. Don't forget, pupil size changes are relatively slow. In this subset of data where we have 20 participants, each of which has 240 trials, and we are looking at 2,000 milliseconds of data, if we did a sample report at this point and just output raw sample data, we would have millions and millions of rows. What we can do is kind of piggyback onto a report that was really meant for use with visual world type data. It's meant to bin gaze data, but it will also bin pupil data, which is very, very useful. So we're going to do a time course binning analysis. And again, you've seen that I've chosen my experimental variables, and one for who the participant is, one for what trial we're on, one for a bin because we're going to get a row per bin in this report, and the variable of interest is the average pupil size in that bin. And we can choose our bin interval, so here I've chosen 20 milliseconds, but you could easily do 50 or 100 even, to sort of radically reduce your data. And if you run that report, you're still going to end up with a file that has, I think this one has about 500,000 lines in it, but it's much more manageable than having all of those lines that you'd have if you had pupil size per sample, for example. So if you run that report, which I'm not going to run because I have run it already, you will get a time series report. And here's one I prepared earlier. If we go over to the raw data, that's the trial report. Where is my time series report? It must be that one. Sorry. There it is. You will see that there is my average pupil bin, and for each participant we have 100 bins, 0 to 99, because we have 2,000 milliseconds worth of data. So 0 to 99 for each participant. And in each bin, assuming that there wasn't a blink, for example, we have an average pupil size. And again, we can do our tricks with pivot tables. This time I'm simply plotting bin index rather than per participant. And if we take our average pupil size, go away, average pupil size bin, don't want a count, we want an average. Hopefully you can see somewhere down the bottom we have plotted it out. It's unhelpfully lost all of the colours. I'm not sure why that is. It's also rescaled it very uselessly. Let's put a minimum in of more like 1,000. And we can see that there's a clear difference. But what you can see most obviously is this. This is when the face or the fractal appears here. There's a delay, about 10 bins, so that's 200 milliseconds, when nothing's really happening. And then you get this luminance response, and it's massive. So there's a huge reduction, constriction in pupil area. But what's interesting, if I could get the colors back, is that there's a difference here. That's actually a difference between the angry faces and I think the fractal faces. And we'll redraw it with the color in a minute. But that just shows the importance of, let's do that, insert line. There we go, that's a bit more pretty. Again, it's not done something sensible with the scale, but you can fix that relatively easily by giving it a fixed minimum. And there, yeah, there you can see. So we've by not having an isoluminant stimulus, we've basically made our interpretation somewhat difficult, because rather than, as expected, seeing a bigger dilation for angry or fearful faces compared to fractals, what we've seen is a lesser constriction. In other words, the pupil constricts because a stimuli has appeared on the screen and it has a much higher luminance levels, but it constricts less to these angry versus fearful, happy, neutral faces, or versus the fractals. And you can see that after it's kind of done that response, there is then a very, very stable period afterwards where pupil size is consistently higher for the angry faces compared to the other three. So there's various other tricks that you can do. For example, one of the things I did in R was to simply, I won't go through that now, is create a column that's the same length as my time series report that has the baseline data in it. So you just expand it by 100 for each participant, basically. And then you can do the subtraction. So this is what I've done in this column here. So this diff column is basically people's baseline, which is here, subtracted, sorry, no, it's a difference column. I calculated it in R, pasted it in. If we look at the difference column and use an average of that, so this is now, whoops, that doesn't look very nice, but we can grab those ones. So there we go. You can see now everything's nice and normalized, and we're now looking at change from baseline within a trial, again averaged over hundreds of trials, but for each participant you can see everything starts off, this is immediately after the images appeared, not much happens, then there's this massive reduction, and then this very quickly stabilizes to a situation where there is a clear difference between angry faces. So angry faces are different from all of the other faces, all faces are different from fractals. This is illustrative data. This is not how you should analyze your data necessarily. Obviously, all data is different, but these are just some illustrations of the kinds of things that you can do relatively quickly using Data Viewer to explore your pupil data and then use it, or use that data to do inferential statistics. If you go back, for example, to the data from the time that the faces were on, you could write a little Excel column that would exclude a trial, for example, if the X and Y data were outside of certain limits at the time that the pupil was at its maximum, if maximum pupil size is something that you're reporting. And again, use that as a filter either in Excel to exploring your data or in some stats package to exclude data at a later event. Or you've got these X and Y values, you've got them, you could get them from the time series report, you could output them in the same format, average X and Y, and you could use that as a covariate in some more kind of sophisticated analysis of a more full, if you like, treatment of your pupil data. There are lots and lots of different things that you can do. You can measure the slope. Well, this is upside down because we're looking at the light response really to an image. But if this was a kind of cognitive response, you could measure its slope, the time it takes from when it starts to when it reaches its maximum. All of these things may differ across conditions or manipulations. So at this point, I think that's about it as far as the analysis is concerned. Thanks for listening. I'm going to stop the recording at this point, but if people are still here, you'll feel free to contact me and ask questions, and I will answer that. So I hope this has been useful. If anybody does have any questions about their pupil analysis, I'm always very happy to talk about data, eye tracking data in general, pupil size data in particular, and you can feel free to email me at the address that you'll see posted in the support forums. Okay, thanks a lot.